Chrome pigments are artifacts that form during fixation when tissues are treated with potassium dichromate-based fixatives, for example, Moller's fluid, Others fluid, or Zenker's fluid. These pigments result from the chemical reaction between potassium dichromate and tissue components, particularly during improper washing or dehydration. In terms of formation process, one, we have potassium dichromate, whereby fixatives containing potassium dichromate are used to preserve tissue, particularly for studying certain cell types, for example, adrenal glands or chromaffin cells. So potassium dichromate leaves chromium residues in tissue. Number two, reaction with alcohol. During dehydration, tissues containing chromium residues are exposed to ethanol, specifically an insoluble reduction product is chromium oxide precipitates in the tissue. So this reaction produces the yellow-brown granular deposits known as chrome pigments. Characteristics of chrome pigments: they appear as yellow-brown fine granular deposits in tissue sections. Number two, they are found scattered throughout the tissue or localized in poorly washed areas. Fixation mechanism: one, washing thoroughly. So after fixation with potassium dichromate-based fixatives, tissues must be washed in running water for several hours to remove excess chromium. Number two, avoid prolonged exposure to alcohol, as this minimizes exposure of tissues to ethanol during dehydration if chromium residues are suspected. In case they have been formed, how are they removed? One, we use acid alcohol. So sections or tissues are treated with 1% acid alcohol to dissolve the pigment. Number four is pink disease artifact. Pink disease artifact refers to an artifact seen in histological tissue sections that results from poor fixation. This artifact is associated with tissue autolysis and improper processing, which causes an even staining, especially with eosine during hematoxylin and eosine staining. And this artifact is often a sign of incomplete or delayed fixation. Formation process: one, it will be formed when there is delayed fixation. So tissues not promptly fixed after collection begin to undergo autolysis, which is self-digestion by enzymes. This leads to protein degradation and structural breakdown. Number two, inadequate fixation penetration. So if the fixative does not adequately penetrate tissue, certain areas remain underfixed or unfixed, allowing degradation to continue. Number three, shall have binding of eosine. So during H&E staining, degraded proteins in autolyzed areas exhibit increased binding to eosine. This creates excessively pink-stained regions, giving rise to the pink disease artifact. Why is it that severely autolyzed tissues exhibit increased binding to eosine during histological staining? One, protein denaturation. So during autolysis, proteolytic enzymes break down proteins in tissue. This process exposes more basic amino groups like lysine residues that can bind more readily to eosine as an acidic dye. Number two, pH change. So autolysis can cause localized changes in tissue pH due to the release of cellular down of metabolic byproducts. So a lower pH enhances the interaction between eosine, which is negatively charged, and positively charged tissue components. Number 3, loss of nuclear material. So severe autolysis often results in the degradation of DNA and RNA, so reducing basophilic or hematoxylin-stained structures. So this allows xenophilic components like cytoplasmic proteins and extracellular matrix to become more prominent. There is increased cytoplasmic eosinophilia. Autolysis leads to the collapse of organelles and the release of cytoplasmic proteins, which are eosinophilic. So the abundance of exposed proteins increases eosin binding. So pink disease artifacts, they appear as uneven staining in tissue section, and affected areas appear excessively pink due to strong eosin uptake, and common in thicker tissue samples or poorly processed specimens. Causes can include delayed fixation, so tissues that are not immediately fixed upon removal. Number 2, insufficient fixative volume. So inadequate volume of the fixative relative to the tissue size prevents proper preservation. Number 3, improper tissue handling, like thick tissue blocks or improperly trimmed specimens hinder fixative penetration. Number 4, autolysis, degradation of tissues caused by enzyme or bacterial activity in poorly preserved samples. Prevention can include prompt fixation, whereby tissues are immersed in the fixative immediately after collection to prevent autolysis. Adequate fixative volume, whereby the volume of the fixative should be at least 10 to 20 times the volume of the tissue. Number 3, proper tissue trimming or cutting. Trim tissues to 3 to 5 millimeters thickness to ensure complete fixative penetration. Number 4, use high quality fixative, whereby you must employ standard fixatives like neutral buffered formalin at 10% for consistent fixation results. Additionally, we can use 2% acetic acid or 1% HCl. So the 2% acetic acid is added to the fixative, or the 1% HCl is added to the absolute ethanol used to dehydrate sections. Addition of acetic acid lowers the pH of ethanol. So a lower pH prevents the precipitation of tissue components, majorly proteins and phospholipids, that can otherwise occur in more alkaline or neutral ethanol solution. And this stabilizes the tissue components and ensures proper preservation during dehydration process. Number 2, hydration effect. Acetic acid and HCl enhances the dehydration process by ensuring proteins and other cellular materials do not cross-link or coagulate abnormally due to unfavorable pH conditions, preserving tissue integrity. Acidified ethanol ensures that tissue morphology and chemical properties remain intact, minimizing the risk of artifacts that could interfere with microscopic evaluation. Correction in case these pigments have been formed. 1, we must appreciate that pink disease artifacts cannot be removed once they form. So why? Because they are formed as a result of structural changes in tissues that cannot be reversed. So the only solution is prevention through proper fixation techniques. Lastly, what could be the general effects of artifacts on diagnosis? 1, these artifacts can lead to misinterpretation. So artifacts can resemble pathological changes, e.g. deposits, pigments, or abnormal staining patterns, leading to incorrect diagnosis. Number 2, masking of pathology. Pigments or artifacts can obscure tissue details, preventing identification of true abnormalities like necrosis, fibrosis, or malignancy. Number 3, alteration of staining. Many artifacts interfere with routine staining methods, for example, hematoxylin and eosin staining method, causing inconsistent or even staining patterns. Thank you.